Hi, as RJ said, my name is Maria Davis, and I have the extraordinary opportunity of being a Dimensions Fellow in Research at the University of Iowa. This past summer, I had the wonderful opportunity of being able to work in 2 different labs at the University of Iowa Hospitals and Clinics and the Carver College of Medicine. I was able to work at both of the University of Iowa's research sites as I worked for the Iowa Inflammation Program in the Suderwalla lab with Dr. Sophie Jolie, who studies the innate immune system or the initial response to invading microorganisms. This was on the Oakdale campus, which is in Coralville. And I also was able to work on the main campus in the Department of Pediatrics in Dr. Martine Dunwald's lab under her direction as she studies skin and epidermal development and regeneration. So as I said, I had the wonderful opportunity of working in 2 different labs. It was a great experience to work with in an interdisciplinary role. As I did this under the 2 researchers, I was able to assess the role, beneficial or detrimental, of a specific innate immune molecule in the wound healing process. My time over the course of my 8 weeks could be split into 3 different categories. I began with learning the required lab background techniques and research by reading literature as learning directly from Martine and Sophie. And then I carried out and studied the in vivo model of our project, which was working directly with the mice, wounding and then watching their healing process. And then I also studied and carried out the in vitro model, which was working with isolated skin cells from these mice to see what their basic behavior was. So more specifically, I worked on the investigation of NLRP10, NOP10, which is the nucleotide binding domain, leucine-rich repeat protein 10, and so I won't ever repeat that again. But there you go, that's right now. So as I said, I studied this receptor, and it is in a family of receptors that is known to produce and secrete pro-inflammatory cytokines, and cytokines are molecules that are secreted by the body to protect you against inflammation, infection, and trauma. And so also NLRP10 receptor is also known to be expressed in epithelial cells of the body. And so we studied them as they were expressed in, we also know that they're expressed in keratinocytes, which are epithelial cells of the skin. And so therefore, we had the hypothesis that NLRP10 was involved in the wound healing. And so during my time, we studied wild type, which are normal mice, versus this NLRP10 knockout mice, which are the mice that do not contain the NLRP10. Therefore, during my time, I followed up on a pilot study that was previously done by Sophie and Martine. And this previous study assessed the appearance of the wounds from the macroscopic level, which was taking pictures of these wounds daily after they were wounded. And then the area of these wounds were measured, and then they were compared to see what the overall % closure was over these 7 days. The initial study showed that the wounds of the knockout mice, the ones that don't have the gene, actually were closing faster than those of the wild type mice. Therefore, it was possibly believed that NOP10 was either slowing the process or could inhibit the closure of these wounds. So my study then was to follow up on these. This is actually important as inflammation cells are important to your body as they are used to clean the wound before it initially closes. So if you have closure of the wound before it's officially done, you have at risk for infection or extreme scarring, other things like that. So this is important, especially in patients that are at risk for having wounds, such as diabetic patients or burn victims. And so investigating this can help us maybe determine a special treatment or something along those lines by understanding its role. So during my time, I worked with the mice, wounding them and following them throughout the entire process, taking pictures of them daily and then analyzing them macroscopically. Also, I took these wounds when we were done, embedded them in paraffin, and then they were going to be sectioned, which is also known as morphometric analysis. This process is kind of like cutting meat at the deli department into really thin slices, and then you assess them to see different things that are involved throughout this time. Also, as I said, I did in vitro work, so I worked with tissue cultures of the wild-type cells. Unfortunately, we didn't have the knockout mice to use at this time, but I was able to prepare successfully tissue cultures and then run different tests on them. One of these tests was the scratch assay. This is an example of a scratch assay. So these are the cells before. We grew them in a dish. And then this was directly after we did the scratch assay, which essentially is like making a hole in them and simulating a wound-like situation, and then watching them progress as they migrate back together. So this is after time of 12 hours and time of 24. So as you can see, these cells are moving back together, even though the wound is not completely healed in this situation. So science takes a really long time, and unfortunately, during my short 8 weeks, I wasn't able to completely finish my project. But at this time, we still believe that the wounds of these NAP10 mice are initially closing faster. Also, I was able to completely embed and prepare all of the wounds to be morphometrically analyzed in the future. And I also finished the in vitro work of the wild-type mice that can be used for future comparison when we have the knockout mice done and ready. These two things will give us an overall idea of what the wound healing process is like underneath the surface of the cells. And so we get an overall picture of what's going on. Also, it'll give us an idea of what the role of the NAP10 is in these mice. This is an example of the wound closure analysis that was done. This is actually taken from the pilot study, so not from what I did. But as you can tell, the NAP10 definitely initially closed faster as the big drop in day 1 compared to the wild type, which is the darker line at the top. I've done research before, but this was definitely the best experience that I've had so far. It was definitely a great learning endeavor throughout the course of my 8 weeks. Intellectually, I was challenged to learn about the different fields of study, as I was not prepared or had no really previous knowledge in the inflammatory system or skin. Also, I learned different laboratory techniques that can be used in my future, and I built upon those that I began here at Cornell. I took my time across the wet bench and also improved my skills in animal laboratory skills as well. Professionally and personally, I also grew. I learned how to independently structure my time with the help of my 2 wonderful mentors and produce scientific work in a timely fashion. Also, I learned the extreme importance of learning to read literature, keeping organized notes, and asking questions. This fellowship definitely had an impact on my life. Academically, I had firsthand experience in a research setting, which was wonderful to build upon that I began in the Cornell classroom. I was also challenged daily to think critically about what I was doing and why I was doing it and what the importance of that was to my project and beyond. Professionally, I gained an opportunity to participate in teamwork, working with other colleagues and the different people in our lab. And I also gained insight into conveying and exchanging ideas with those that I've worked in and with. In the future, this has helped me to further ignite my interest in the medical world, as medicine and research are so intrinsically intertwined. And it also helped foster self-confidence in both my scientific abilities and the future career planning. So I'd like to give a big thanks to the Dimensions program for funding my wonderful opportunity. My site mentors who are both with me today, Dr. Sophie Jolie and Dr. Martine Dunwald. Other lab members, Dr. Faz Suderwalla, Dr. Jeff Murray, and Dr. Leah Biggs, who helped me along my way. And my faculty sponsor, Professor Craig Tepper, for all his help as well. Thank you very much.